much the organisers of this, of HASDAR. Thank you very much for inviting me, and it's been an absolute pleasure to come here and meet all the fine researchers here. Thank you. So my talk today is about, my talk today is really about an Australian perspective of the latest research that we are undertaking in Australia. So, okay, so I put this map of Australia up here just to demonstrate a few things between my country and perhaps your country. So Australia is really a huge continent, but the amount of land that we can actually use is very small. And so we have our major cities all around the coast, and here, all this area here is really mostly desert. And 90% of the people, if not more, live in these capital cities. And the country is divided up into 6 states, with Western Australia and Perth as the capital city, and Adelaide, which is dear to my heart and where Kien has done his PhD, is in South Australia, the capital city. Melbourne is the capital city of Victoria, which is very small. And then Sydney, where I also work, is the capital city of New South Wales. Brisbane, capital city of Queensland, which goes all the way up here. And then Darwin and Canberra, which are capital Darwin is a capital city of Northern Territory. It's not a state, it's a territory. And Canberra is the capital city of ACT, which is Australian Capital Territory, but also is the capital city of Australia. So we have a population of 25.7 million people, so not many in comparison to Vietnam. And births per year, 294,000, and ART births per year is 37,600. So very different from the results that you have here. So the contents today, I'll be discussing the history and trends of ART in Australia, a little bit about in vitro maturation, and mainly I want to talk about mitochondrial donation and mitochondrial supplementation. So if I start on the history and trends of in ART. So I start really research into IVF and ART started a long time before 1970, but mainly in animals. So, well, all in animals, mainly in rodents and rabbits, where they were able to successfully produce live offspring. But in 1970, Steptoe and Edwards from the UK, Steptoe was a gynecologist, Edwards was a physiologist, got together based on the studies done in animals and performed the first IVF with human oocytes. So in vitro, just first human oocytes getting around to about blastocyst stage. In 1978, I think the first and second IVF babies were born, again, due to Steptoe and Edwards in the UK. In 1980, in Melbourne, and people that I know and had studied also in Adelaide, produced the third IVF baby in the world. So we feel that for such a small population, we have really made a significant advance. After that, in 1980, a year later, twins were born in Melbourne. Then in 1982, triplets were born in Adelaide. And then 1983, first frozen embryo birth in Melbourne. And then by 1984, there was quadruplets born in Melbourne and a total of 365 IVF babies born in Australia. So within a very small amount of time, we made some very significant global inroads into ART and IVF technology with quite a lot of success. So in 1986, before I jump too far ahead, the first baby born from a frozen egg was done in Melbourne with Alan Trounsden, and very few since. So after 1986, not much happened in Australia for quite some time. A lot of animal work had taken place, but very little real developments in IVF in that time in Australia due to the regulation and ethics development. So committees were established and funding towards IVF was not given until all the regulations were worked out. So IVF in Australia, just a few statistics here. Approximately 1 in 20 babies before COVID were produced or born due to IVF. Now it's dropped off some. It's picking up again, and it's around 1 in 28 babies in, I think that was 2020. So one in 6 couples are believed to have some form of infertility. 1 in 18 babies are born from ART, and 1 in 10 babies are born in women after 35 years of age. So regulation and ethics, Australia, I suppose, is one of the highest regulated countries as far in the early days, especially with regard to ART. Following the first baby, the Victorian government, so most of this happened in Victoria first, because that's where the first baby was born, and a committee was established in 1982 to investigate the social and ethical and legal issues surrounding IVF procedures in Victoria. In Australia, we have local governments in every state, and the laws in states regarding ART, but regarding most things, are different depending on which state of Australia you live in. So in 1987, the Fertility Society of Australia, FSA, which is still running, of course, set up the Reproductive Technology Accreditation Committee to implement a national accreditation scheme for ART clinics. So that began very early on. Victoria was the first jurisdiction in the world to introduce comprehensive legislation to regulate the use of ART in 1988. So the 5 guiding principles that came from this committee was that the welfare and interests of persons born or to be born as a result of procedures are of paramount importance. And at no time should any of the treatment procedures should be used for the purpose of exploiting in trade or otherwise of reproductive capabilities of men or women, children born as a result of the treatment procedures. Children also born from donated gametes have the right for to their information or to the information of their genetic parents. So everything is kept, everything is recorded, and when children reach the age of 18, they are allowed to have or permitted to have all the knowledge about their genetic parents. The health and well-being of persons undergoing treatment procedures must be protected at all times, and the persons seeking to undergo treatment procedures must not be discriminated against on the basis of their sexual orientation, their marital status, or race or religion. And now in some states, same-sex couples can have access to IVF technology, and in some places it's around 5% to 10% of ART births. So the national accreditation schemes encourage also the use of single embryo transfer. So if you saw my timeline, there were many multiple births in the early stages. And with multiple births, the babies usually do more poorly. So this is a strongly encouraged thing. We still do 2 embryo transfers, but by far the most of them are single embryo transfers. And the rate of multiple births has dropped from 8.4% in 2008 to 6.5% in 2012. So this resulted in better outcomes, of course, for the mothers and babies. So I put another couple of slides up just to show trends that in Australia, which I am very sure mirror the trends here in Vietnam and elsewhere. So this one here, this is a look at success rates of frozen thawed embryos from 2009 to 2019. And here we have separated these into greater than or equal to 45 years, 40 to 44 years, 35 to 29, and I can't really read that, but 30 to 34 and younger. So on average for these, the success rates have increased from 18.4% in 2009 to 25% in 2019. This side is graphing the success rates from fresh embryos, again categorized towards ages as it is here, and it increased from 23% in 2009 to 25.3% in 2019. So the overall success rates for all embryo transfer has increased from 21.2% live births, of course, in 2010 to 27.9% in 2019. So I've put here probably it's back to 1 in 20 births, and very close to that is now conceived due to ART. So IV success rates, maternal age, of course, around the world is the biggest predictor of IVF success. And IVF at the moment is not able to really address the issue of maternal aging and reduced fertility. But next to, say this, I find this a little bit controversial, but next to maternal age, choice of IVF clinic is the best predictor of IVF success. So you've got maternal age first, and then the choice of clinic. And so I'm going to put up some statistics now with regard to that from Australia. So if we take here age group, maternal age, less than 35, 35 to 42, births per egg retrieval, including later transfer as a percentage. We have the different IVF clinics here, so Genea, Flinders Fertility, so this is in South Australia, Repromed is in South Australia, and Kian and myself have had a lot of interaction with the with Repromed Clinic, Care Fertility in Queensland, and then I've got here the national average. So if you can see here, Janaea in Newcastle, for this one, births per egg retrieval, 64.8%. It almost seems unbelievable, okay? And some of us question it, but these are sort of verifiable data. I didn't put the website here, but you can easily check that. So the next one here is birth per... So what I want to show here, and it's not really to support Janaea in any way. I have nothing to do with Janaea. I have a little to do with the Repromed, and I know the people, of course, running Flinders Fertility, but I want to say this is the difference. So national average 44%, 44.4, 64.8. Likewise, for births per individual treatment attempt, 43.4%, 31.6. Down here, births per embryo transferred, 46.1 for this clinic here in Newcastle. Newcastle is actually in New South Wales and is a very small city, especially in Vietnam standards. But 46.1 and 33 as an average. So the variability between the success of clinics is true. It is the next determinant of sex after maternal age. So I'm going to talk about IVM, in vitro maturation. But I had a lot of slides to talk about this because Australia has been involved in IVM for quite some time. But I have taken most of them out because my talk went way too long. And, you know, with the studies at Ho Chi Minh City, there is a lot of information about IVM already in Vietnam. So I don't think I'd be actually telling you very much at all. But we just go to the next slide. So IVM maturation history, again, Trounsden from Melbourne, Alan Trounsden, he is very well known globally, of course, in this area, but very, very well known in Australia. And when I was doing my honours and PhD, we all knew Alan Trounsden, and we were always worried about his questions at conferences. So, we come here, the number of the number of pregnancies in the early days. I got a live birth from Korea in 1991, 1994, pregnancies. But as we go down, Mikkelsen, Chan, Chin, these are the main players with IVM, and the estimated IVM babies, if you like, is greater than 5,000 from 2013. So I dare say, I'm sorry I haven't got any statistics more recent than that, but it must be 10 times that now. So IVM, currently the most promising development is Kappa IVM, which Ho Chi Minh City has been involved in with collaboration with Robert Gilchrist in New South Wales, it's also in Sydney. Rob Gilchrist started off his postdoc years in Adelaide in our department, and we all know him very well. But in collaboration with scientists from Belgium, he is investigating Kappa IVM with studies in Vietnam from Ho Chi Minh, as I mentioned, have established and validated Kappa IVM. Currently, it is being used in Australia in a fertility research centre in the Royal Hospital for Women in Sydney, with Rob Gilchrist, keenly interested, of course, in that. He's a scientist, of course, but also with Bill Ledger, which a lot of people here would know him definitely. So I put this slide up to introduce the concept of getting people to think about mitochondria. Mitochondria are organelles. I'll show a couple of slides, but they're organelles in all cells. And of course, they provide the energy for the cells to live and the energy for us to exist, of course, and produce ATP and oxidation and respiration, etc. So just quickly, the reason why I put this up, and it's not a great slide, but you can see that these are mitochondrial DNA. So each mitochondria has a single strand, a circular loop of DNA. And we always refer to it as mitochondrial DNA and copy number. And what we know is that this copy number, or the amount of mitochondria, does change and varies due to stage of oocyte development. And therefore, as you would expect, the bigger the cell, the more ATP and energy is required. So I'll show more about this a bit later, but all I want you to understand is that the values here are very important for oocyte quality, okay? Okay, so we're just going to start with mitochondrial donation. Okay, so in Australia, I have alluded to the fact that we are highly regulated for everything to do with medicine, really. I think that is the way to be, but I'll just say eligible women, so just recently eligible women in Australia with a diagnosis of mitochondrial disease may have access to mitochondrial donation under amendments to the Human Embryos Act that was established in 2002, and the prohibition of human cloning for reproduction again in 2002, which took effect on just a couple of months ago, on the 2nd of October in 2022. So as soon as that legislation actually was passed, we now have money available to do trials in mitochondrial donation. So we are not at the stage, we've only had 2 months of permission, so we're not at the stage of producing babies or there, but we have done a lot of animal work associated with mitochondrial donation. So yes, recent changes, right. Has meant that grants are now available for clinical trials and cohort studies to evaluate mitochondrial donation to be used clinically in Australia. And this is similar to what has been going on for a number of years now in the UK. So there is not much information in the literature about babies born from mitochondria. There is some, but and the UK have produced a number of babies using pronuclear transfer, so I will talk about that. So the goal of mitochondrial donation is to prevent second-generation transmission of mitochondrial disease. And mitochondrial DNA is passed on through the maternal line. So the main techniques to avoid offspring developing a mitochondrial disease rely on the replacement of defective mitochondria with non-pathogenic mitochondria from donor zygotes or oocytes. So zygotes or oocytes. Currently under investigation in Australia are pronuclear transfer and metaphase 2 spindle transfer. So just to briefly talk about mitochondrial disease, which you may be very familiar with. It's a group of disorders caused by dysfunctional mitochondria, usually due to a mutation, either in the nuclear genome or in the mitochondrial genome itself. And mitochondrial diseases are sometimes caused by mutations, about 15% of the time, in the mitochondrial DNA itself that affect mitochondrial function. Other mitochondrial diseases are caused, as I said, in the nuclear DNA, whose gene products are imported to the mitochondria. Incidence of mitochondrial disease in Australia is 1 in 2,500 and can be diagnosed at any age. Some of the diseases are very severe and often results in death of a child very early on. So the history of mitochondrial donation, sort of came about from the human mitochondrial genome being sequenced in the UK in 1981. There's been nuclear transplantation in mice. In Switzerland, mutations in mitochondrial disease have been linked to diseases of the muscle, myopathies. In 1988, mutations in mitochondrial DNA linked to Leber's optic neuropathy in the US. Protocols. So again, protocols and regulatory policy development began quite early on in the UK. Gene replacement in primate offspring and embryonic stem cells occurred in 2009. Pronuclear transfer in human embryos it took place in Newcastle upon Tyne in the UK. And a baby boy was born from MST, or the metaphase 2 spindle transfer, in the US in 2017. So I had to give us a little bit of a promotion here in Adelaide. So what we have been researching on for the last, probably the last 10 years, but for me, the last 5 years with Professor Just St. John, that's pronounced, or St. John, but it's pronounced St. John, and me in Adelaide. So this is mitochondrial supplementation instead of mitochondrial donation. So just very briefly, this is a mitochondria, and we have some mitochondrial DNA, circular DNA here. They mediate a number of key cellular processes, ATP, as I was saying before, DNA methylation, and also steroidogenesis. So possess 2 to 10 copies of mitochondrial DNA encoding. So this DNA here only encodes 13 subunits of the electron transport chain, which generates the vast majority of cellular ATP through oxidative phosphorylation, which I'm sure you're familiar with. The oocyte is the richest cell in the body in mitochondria. It's a big cell, and it's the richest cell in the mitochondria, and it depends largely on these mitochondria to acquire competence for fertilization and early embryonic development. So I'll just talk about these two techniques. So pronuclear transfer is when you have a fertilized egg and the pronuclea are formed, and this is in the donor egg here. And you take out the pronuclei. I've got another photo of that in a minute. And then the recipient oocyte, again fertilized, and you take out the pronuclei here from the recipient oocyte, and then you put this donor pronuclei into the affected, the healthy egg. So what you're taking out is the parental DNA, if you like, and putting that into a recipient egg that has had the pronuclei taken out. And here's the reconstituted zygote with the recipient DNA and the healthy donor mitochondria. I hope that's clear. Here's a micrograph of the process. Here we've got the pronuclei. It comes out here and then, of course, goes back into the recipient egg. Okay. Metaphase 2 spindle transfer. It's a similar process, but it happens before fertilization. Again, here we have the donor oocyte, so healthy mitochondrial DNA, mitochondria, or mitochondrial DNA we usually refer to all the time instead of mitochondria, and that is colored green, and that is taken out. The donor spindle is taken out and replaced with the affected or the potential parent's mother's metaphase spindle, and that is replaced in here. A sperm is fertilization takes place at the same time with the delivery of a single sperm, and away we go with our reconstituted zygote. So healthy nuclear DNA and also healthy mitochondria. So with these two processes of taking the pronuclei and also taking out metaphase 2 spindle, of course there is some carryover. So there is some cytoplasm with mitochondria that is also taken out as well. And this is the holy grail of this technique, is to reduce the amount of mutant, if you like, mitochondria being transferred across. Okay, so and what that's called is heteroplasmic. So if there is any transfer, so when you take out the spindle, when you take out the spindle here, if there is any transfer of mitochondria, depending how much, then of course the zygote will be heteroplasmic. So it would contain some of the green, or most of the green, but also may contain some of the red, yeah? So we call this carryover, and if there's significant carryover, then it's heteroplasmic. So you've got two populations of mitochondria. It doesn't seem to be a huge problem from studies that we have done in Adelaide, but it is something to be considered when you're thinking yourselves about when you want to do mitochondrial donation, which one that you will pick, okay? This is just a micrograph again of the spindle coming out here and the process of producing the reconstituted zygote. So pros and cons, as I said, from our experience, there is not a lot of data where you can weigh this up clearly, okay? And hopefully the trials that we will be doing will investigate this as well. I do want to say that pronuclear transfer so this again is Adelaide experience, right? We do pronuclear transfers in pig, using the pig model, and we tend to think that it's technically easier, but this may be due to experience. it has low mitochondrial carryover, less than 2% mitochondrial DNA. And we can measure that, of course, because we can do PCR for mitochondrial DNA, and we can do quantitative PCR and measure the carryover. And it is regarded less than 2% it's not clinically significant. Metaphase 2, technically a little more difficult. Again, this is Adelaide experience. I have no data to show you because we just have not done enough. But again, there is low mitochondrial carryover, but for us, there was a little more than pronuclear transfer. But this is not significant also. So please don't look at this as a negative for metaphase 2 spindle donation. So clinical trials are now approved in Australia, as I said. The research is expected to be underway in 2023 with NHMRC applications being evaluated. NHMRC is our funding body for all for most of the medical research done in Australia. So I know I know lots of research groups, but 2 research groups that have currently been researching in this area, one in Sydney and one in Adelaide. And hopefully they will both be funded. So mitochondrial supplementation. So this is where you either get donor mitochondria and you supplement the oocyte, or you use autologous mitochondria. So it could be mitochondria from, if you superovulate, picked up a number of eggs. You could use eggs that didn't fertilize. You could use those mitochondria and you could pool them. Then it would be autologous. So it would be homoplasmic, okay? But this is supplementation we're talking about now here, not nuclear donation. So we have done quite a bit of work in this, and I'm not sure how much time I've got left, but I will talk quickly through this. Again, we talk about the role of mitochondria in oocyte function. And mitochondria provide the number of mitochondria DNA, copy number, provides a critical indicator of oocyte quality. And it's important, again, I mentioned this before, but it's important for fertilization and development into viable offspring. So quality compromised oocytes are correlated with infertility. We all know that. Developmental disorders, reduced blastocell number, and embryo loss. And we maintain, and it's our hypothesis, that mitochondrial dysfunction plays a significant role in all this. And increasingly, women are affected by metabolic disorders such as diabetes, obesity, oocyte aging, and there is thinking, of course, IVF, clinics to overcome these problems. And our research project has our research project has a relevance to ART in investigating how we can increase. So women that have recurrent spontaneous abortions, miscarriages, it may be due to something to do with the mitochondrial level in their oocytes. And we have some evidence to support that. So again, I've got a slide here just to emphasize the importance of mitochondria in oocyte development and oocyte and embryo development. So we know that it starts off, as I showed you in my previous slide, that early in oocyte development, the amount of mitochondrial DNA copy number is around 200. 200 copy number of 200. And as the oocyte develops and the oocyte grows, so does the reproduction of these mitochondria, and it rapidly grows. And it goes up to about 150,000 just prior to fertilization, and at fertilization it's over 200,000 copies within the oocyte. If it does not go up with this way here and goes this way, doesn't meet the threshold, then you have a problem with pre-implantation development, and the oocyte either doesn't fertilize or doesn't develop past a particular stage, which we can't tell you exactly, but that it certainly has implications here. And then just to finish this slide, then we come along here, and normally what happens is after fertilization, there's a sudden drop in mitochondria. And then we come down in the trophectoderm, mitochondria increases, but as we go down, primordial germ cells, for instance, it stays pretty low. And then in different cell types, there's different levels of mitochondria. So it changes between neurons, muscles, heart, and blood. Variability between that in mitochondria number. So this is a system that Professor Just Sinjen in Adelaide has developed, mainly in Monash before he came to Adelaide. So we have metaphase 2 oocyte, roughly greater than 150,000 copies of maternal DNA, and if that is the case and fertilization takes place, the sperm here, then you get a blastocyst and you're likely to get a baby. If you have less than 100,000 copies, which was that threshold, fertilization can take place, but doesn't go any further. So what we do is we try and get, these are all green, right? So we try and get autologous transfer. So, but we have done both. Autologous transfer of mitochondria, and it would be from another oocyte. We use the pig, we collect the ovaries at the abattoir. We can get as many ovaries as we want, practically, or can cope with, and we just, of course, extract the oocytes from the follicles and then go through IVM. Some continue, and some we use to get the mitochondria out for doing these sorts of experiments. So the hypothesis is if we increase the mitochondrial DNA copy number in mitochondrial DNA deficient pig oocytes through, we call it MIXI for mitochondria ICSI procedures, offsprings are healthy and fertile and does not affect the health of the next generation. So that's our hypothesis in our big study that we did a couple of years ago. And the aim was to assess the health, fertility of offspring of these that had received more mitochondrial DNA. So the objectives were to restore developmental competence in IVM oocytes that failed to fertilize. So we have these oocytes and we try and identify which ones are likely to be successful and ones that are not. And hopefully I can demonstrate that to you. And then we do the assessment at birth and offspring to weaning, and then fertility and sexual maturity, all that sort of stuff. So I'll keep going. It's a little bit complicated to talk about it here. So that's me here doing some surgery. That's my daughter who's helping me with my surgery. And this is a pig. Very different to what happens in fertility clinics, I can tell you. Okay, so we got embryos that we developed to day 3, and then we transferred into the pig. Then we to pregnancy. So we work up the pig with FSH and LH, and just before ovulation, which we have a very good idea when ovulation happens in a pig after giving FSH and LH. And then we either have non-pregnant ones, we get pregnant, and then they deliver farrow, and then lactation, weaning, and so on. And we record absolutely everything, like how quickly the piglet stands when it's been born, how quickly it finds the nipple for a drink after birth. We weigh them and weigh them and weigh them and weigh them, and we feed them and feed them and feed them and record absolutely everything, so we know how healthy they are to control counterparts. So again, we do all this. I'll go through a little bit quicker, but we produce 32 piglets, and we assess 16 at maturity, fertility, and then we produced a ton of babies from that as well. Okay, so increasing this, I'll just say this is our procedure. We took out donor oocytes and we produced heteroplasmic ones, so we got green and red, and we got heteroplasmic ones. We transferred them into the pig. We also did that again for the others. So it'll be very quickly about this, but we assessed good quality oocytes from poor quality oocytes. And how we did that is we looked at a pool of oocytes and we stained them with a stain called Brilliant Crescel B stain. Here, it's like methylene blue to the people who have done a little bit of histology from school. But BCB positive have greater than 150,000 copies. So this is how we determined the threshold for the amount of mitochondria that an oocyte should have. And then they progress to metaphase 2 and develop fertilization, after fertilization. Poor quality oocytes, this is the mechanism, but reduce BCB to a colorless state. So these ones that stain blue, it's a very light color blue, but easily to identify. And then they are developmentally incompetent. So again, all this, but I'm going too long, so I'm going to really skip this slide. So this is, can you take one back, please? So this is our results, really. We produced a number of pigs where we had homoplasmic, so these are all green. So these were autologous transfer, so the transfer from the pig, same pig that produced the oocytes that were fertilized. And then we had the heteroplasmic, and we produced a number of piglets, and we raised all them up and compared the health and safety criteria for those pigs. And we found that with heteroplasmic or homoplasmic offspring, the health and safety and development and growth was identical. And this was all checked by veterinarians and well established. We are just about to publish a paper. We've had it back from reviewers, and now we've sent back our comments. So we're waiting to publish this study shortly. Okay. So thank you very much for your attention, and I'd very much like to thank the organisers and the president, Tao Ding Nguyen and Sin Trin. Sorry for my pronunciation, it's terrible. Mrs. Han, Van, is all I know him by, but thank you. And Dr. Tien, who has been looking after me for this trip and took me around on a motorbike, which I was very, very impressed with Hanoi traffic. And of course, my PhD student, Dang Kien Nguyen. I'd like to pass on my grateful acknowledgement of all their help for me. Thank you.
Thank you, Professor Sin O'Leary.